Okay, so thank you so much, firstly, for this opportunity to present my research work. I am Sheetal, and I'm a PhD candidate in Dr. Jonathan Khan's lab at Case Western. So we mainly study HIV infection of both the brain and the periphery, but today I'll be talking to you about my project, which is based on studying these extrinsic regulators of HIV latency as well as microglia phenotypes using iPSC-derived models that we have established. So there has been sufficient evidence today showing that the human immunodeficiency virus that causes AIDS can actually get into the brain and establish infections. And about 15 to 60% of HIV positive individuals who are receiving antiretroviral therapy still experience different degrees of HIV associated neurocognitive disorders. And these are collectively called as the HAND syndrome. So hand can actually range from an asymptomatic condition to severe forms of dementia. But fortunately, with antiretroviral treatment, we are able to see reduced proportions of severe dementia, but these individuals still face mild forms of neurocognitive disorders. So the exact molecular mechanisms of and the reasons of these mild neurocognitive disorders aren't fully understood. So before I go into what we exactly study, looking at the HIV's life cycle, the virus is a retrovirus consisting of these RNA genome. So they can infect the host cells by a receptor-mediated mechanism. And once they infect, they fuse with the host cell membrane and quickly inject their genetic material, which is this single-strand RNA molecule. And the RNA can very quickly get converted to double-stranded DNA. And the virus actually brings along with it a lot of these host restriction factors. So it is able to evade a lot of these innate signaling mechanisms. So the viral DNA then gets integrated into the host genome. And this actually forms a very critical aspect of the virus life cycle, because at this stage, the virus can become transcriptionally silent, or it can become latent within the host cell. And the period of latency can vary in each individual. So, under certain circumstances, which I'll be talking about later, the virus can reactivate and make more of viral RNA copies, producing more viral proteins and package new viral particles, which then get released and start this new round of replication. So, what our group has been studying over the years are these mechanisms of HIV latency and regulators, both cell intrinsic as well as extrinsic regulators of HIV latency. So, a major challenge with HIV cure is that the virus can rebound from these latently infected cells when the treatment is interrupted, or when these HIV positive individuals encounter certain opportunistic infections, or have systemic inflammation. So, looking more closely at how the viral transcription is regulated, our previous work have shown that the virus can actually become latent in microglia cells. And this is often characterized by a lot of these epigenetic factors or blocks that are introduced to the viral promoter region. So this can actually completely silence the viral expression. The promoter also is heavily dependent on inflammation. So it has binding sites for NF-kappa B as well as IRF3. So in a latently infected cell, we see NF-kappa B sequestered in the cytoplasm. Uh, that's also preventing the virus from getting transcribed. But when we expose microglia to these pro-inflammatory activators like TNF-alpha, which can activate the NF-kappa B pathway, the viral so these factors can bind to the viral promoter region, which will then reactivate them from these latent or transcriptionally repressed state. So thus the latency that is imposed by these epigenetic factors can be reversed by inflammation. So what we have previously seen in our lab is that the brain environment consisting of neurons and astrocytes can actually contribute to latency in microglia. And this is mainly because neurons have an innate ability to suppress this excessive microglial inflammatory state by targeting the NF-kappa B pathways. But we have also seen that when we expose microglia to TNF alpha, that quickly activates the virus, and the viral proteins that are produced in combination with a bunch of different pro-inflammatory cytokines can induce neuron damage. And this sets off a neurotoxic cycle where the damaged neurons are secreting more DAMs and ATPs, which will further hyperactivate microglia. So, our hypothesis that we have been working on is that the periodic emergence of HIV from these latent states may contribute to the development of these neurocognitive disorders. So, what I was mainly interested in was understanding these microglia phenotypic states and how they correlate with HIV being at this latent stage versus when HIV is fully activated, compared to when there is no infection or when there are these bystander cells. And I was also interested at looking at certain specific signals from the brain environment and how they regulate HIV latency. And finally, if we can identify any early markers that correlate with microglia being in this more hyperactive stage. So, to answer these questions, we wanted a robust microglia cell model um, that can be used to mimic these early stages of infection. And since HIV infects human cells, we wanted a human cell model that could be used in vitro. So we started off by making microglia from these induced pluripotent stem cells. And here we are using the Breton-Jones protocol to make microglia using these different soluble factors secreted by neurons or astrocytes. And using this method, we were able to see increased microglial specific markers like P2Y12 of fractal kind come up and we also verified this by flow cytometry and immunostaining. So then we asked the question of how do these microglia phenotypic changes correlate with HIV being this latent or activated stage. So we started off by giving or infecting these iPSC microglia with the HIV virus. So these viruses had an envelope region which and a lot of these accessory proteins. But also they had a truncation in the GAG sequence, which means that they will not be able to package new virus. This was mainly done so we could contain infections in the lab. And the viral backbone also has GFP region, so we could monitor for these infected cells. And in order to detect latent cells, which will not have any GFP expression, we used TNF alpha to activate the NF-kappa B pathway, which will further bind to HIV promoter regions and further activate the virus, the latent virus. So we saw that in, uh, in, in iPSC models that when we infect the cells, we see some fraction of cells already expressing GFP, meaning that they already had active viral replication going on. But when we activate the cells with TNF alpha, we see a twofold increase in the levels of GFP. And so this indicated to us that even at baseline conditions, we have a fraction of cells that are active, but a fraction of cells that are also latent. And these latent fractions could be activated with TNF alpha. So we asked how we can dissect this mixed population. So we could phenotype cells that are at latency versus the cells that are having this fully activated virus. And to do that, we performed a single cell RNA sequencing, and these UMAPs show us these nice segregation between the uninfected and the HIV infected groups. And when we specifically looked at the HIV transcriptional or the HIV RNA level, we saw that upon infection, we get a small population of cells that have very low levels of viral RNA. But when we expose these cells to TNF alpha, this population now moves upwards with majority of the cells having really high levels of viral RNA copies. So we took this fraction to be a functionally latent group. Uh, and a- and to look at these populations much more closely, we performed certain bioinformatic analysis to section these populations and look at these populations that have very low levels of viral RNA copies that we took to be the latent fraction, um, and the P2 population which had an intermediate level, but the P3 population which had the fully blown out viral RNA. So so the goal with our project was to understand these microglial states, and one way to do that was the antibody sequencing. And so we were able to acquire certain uh, antibodies that were specific to certain microglia markers. Even though we were limited by some markers, we were able to see or correlate these antibodies to microglia states which are at surveillance or performing these FC-mediated responses or having an antigen presentation phenotype or an inflammatory immune phenotype. So, so after performing the antibody sequencing, we were able to now correlate our HIV RNA levels to the protein levels, specifically the microglia surface proteins. And so performing these analysis, we were able to see that when the virus was fully activated in the P3 population, the levels of certain microglia co-stimulatory molecules like CD80 and Fc receptors like 32, 16, and the phagocytosis CD11B receptors were actually enriched in the P3 population compared to our baseline P0 or P1. Um, so we also looked at some antigen presentation receptors that are required for viral recognition, such as HLA-DR and CD154, which were ironically actually downregulated when the virus was fully active. And this actually confirmed a lot of previous observations that HIV is able to evade immune recognition mechanisms, especially the HLA-DR mediated mechanism. So this was really reassuring and interesting to observe in our model. And we were also able to see that certain receptors like CD163, which are actually very low in microglia, were enriched in the P1 population when the virus was latent versus the P3 population. And looking at some of like the microglia genes, we were able to see that genes required for microglial homeostatic or like the surveillance were actually downregulated with active HIV, indicating that these cells may be highly dysregulated. So these initial experiments that we performed showed us that the HIV expression level can correlate with microglial hyperactive states. So our next question was to see um, what are the contribution of extrinsic factors. And obviously we know that microglia do not exist in isolation, so it is critical that we understand what neurons or astrocytes can do to the HIV expression as well as the microglia phenotypes. So to start with, um, we treated the microglia with these different uh, neuron and astrocyte secreted factors and monitored for microglia markers that will or microglia surface markers that will that play an important role in the immune response. So when we treated our cells with TNF alpha and compared them to these conditions which had TGF beta and CD200 and fractalkine, we were able to see that certain surface markers like HLA-DR or the CD45 and CD80 were all downregulated in the presence of these neuron and astrocyte factors compared to the TNF alpha treated conditions. And this indicated to us that the neurons and astrocytes are able to modulate the microglia reactive states, especially the inflammatory status. So, to understand how the physical presence of neurons itself can contribute to this effect, we started off by obtaining iPSC-derived cortical neurons. And for this, we made use of a iPSC line that had an neuroglin 2 transgene inserted at their safe harbor loci. So, using this method, we were able to obtain cortical neurons uh, by 2 weeks of differentiations. And this protocol also gave us glutamatergic excitatory neurons. So next we established these co-cultures with our neurons and compared HIV expression in the presence of neurons and microglia as monocultures. And these showed us that in the presence of neurons, HIV expression were reduced. And when we quantified our results, we were able to see almost a 50% reduction in the levels of HIV GFP expression in the presence of neurons when compared to microglia, which were present in isolation or as monocultures, indicating that the presence of neurons will further suppress the HIV expression in microglia. Um, so uh, here, even though the GFP was a good indicator, it might sometimes be leaky because of the half-life of this protein. So we were looking at more sensitive methods by which we could assay the HIV expression in neurons in the presence of neurons. Uh, so we looked at HIV TAT protein expression. So like I mentioned initially that when the virus is fully silenced, uh, there are certain epigenetic blocks, and so the viral protein that is required for this transcriptional initiation process, uh, that is called TAT, is actually um, falls below a particular threshold. So it becomes undetectable at this latent state. But when the virus is fully active, the levels of HIV TAT expression goes up, and TAT actually auto-regulates itself. So the expression continues to be up of when the virus is fully active. So we looked at HIV TAT expression in our co-cultures and compared them with our monocultures, and we were able to see that the levels of TAT were drastically reduced when we put them together with the neurons compared to when they were present as compared to when the microglia was present as monocultures, again indicating that the neurons have the ability to suppress this HIV TAT. So another interesting observation was how these morphology of microglia were altered in the presence of neurons. So when we have microglia as monocultures, they are mostly amoeboidic with very few processes. But when we put them together with the neurons, especially with these in these iPSC-derived cells, we were able to see that uh, the overall surface area of microglia increased, and they also started to have these processes, and they could be classified to be in a slightly bushier state rather than a more ramified. But in the presence of HIV infection, and especially when we look at these TAT positive cells, we were able to see that when the virus was fully active, the microglia were mostly amoeboidic, indicating that they were at a much higher inflammatory state. So apart from just the neurons and the neurons, we were also interested to further extend this model so we could include all other brain cell types and also use that as a model for HIV latency in the brain. So these are still at primitive stages, but we started off by building these cerebral organoids, which are essentially miniature versions of the brain. So with the help of our collaborators at the Department of Neuroscience at here at CASE, we made these cerebral organoids from iPSCs using neural progenitors that were grown as embryoid bodies. Uh, and these neural progenitors do not give rise to microglia, so we added these primitive microglia precursor cells, which are which were positive for 235, but we allowed for the co-development of this precursor microglia-like cell with the neural progenitors, so that the microglia could differentiate in the presence of neurons and other cell types. And we built these microglia containing cerebral organoids. So looking at these organoids at um, even at day 14, we were able to see IBA1 positive cells beginning to colonize the organoids. And there are a lot of cells at the periphery of the organoid at this point. But by day 25, we lost a lot of cells, but then they became more settled, and we were able to see a much more uniform distribution of microglia in the organoid. And these microglia cells also began to have certain very tiny processes. And we also looked at other cell types in these organoids, like the SOX1 positive or the PAX6 positive, the neural precursor cells. And they were also MAP2 positive neurons that could be seen at this particular stage of differentiation in the organoids. But to get a more comprehensive view of what kind of cell types are in the organoids, we performed a single cell sequencing that helped us identify the major cell types. So when we mapped our data set to the brain atlas, we were able to see that these organoids had a population of neural progenitors, different types of astrocytes, and some oligodendrocytes, and the terminally differentiated excitatory inhibitory neurons. And we did recover a very small fraction of microglia that was integrated into the organoids at this particular stage of differentiation. So, looking at some of the markers that microglia exhibited at this stage, we were able to see fractalkine and IBA1, which is AIF1 gene, show up, and then they were CSF1R that came up. But we still think that this model is still developing, and microglia have a little bit more to go before they become fully mature. So the goal with our cerebral organoid model was to see if we can find a latent virus. So we started off by infecting these organoids with HIV and also treated them with TNF alpha to activate the virus. So when we did that, we were able to see increased levels of HIV TAT expression when we add TNF alpha compared to the unstimulated conditions. And looking at the HIV transcript levels, we were able to see that when these organoids were exposed to different these epigenetic inhibitors, or when they were activated for NF-kappa B and IRF3 pathways, they showed around or almost 60-fold increase in transcription compared to the baseline unstimulated conditions, which were almost zero. And so this indicated to us that this huge fraction of latent cells that could be hiding within the organoids and that were reactivated with these different latency reversal agents.
Sheetal,
So
can you hear
we
me? Excuse me, can you hear me?
yeah,
I'm
I can hear
afraid
you.
we're out of time, so I'm going to have to ask you to wind up, please.
Yes, I okay. Thank you.
Okay, thank
So
you.
this so Yeah, so coming to the end of my slide, so we have seen that HIV expression level correlated with um, microglia hyperactive states, and the neurons were able to suppress STAT expression. And finally, also the organoids can be used to model latency. But the overall goal with our project is to identify these microglia states that are associated with differential HIV expression and look at pathways of these dysregulated cells. So with this, I'd like to thank everybody in Dr. Jonathan Khan's lab, and especially Dr. Khan for guiding me throughout this project, our collaborators, and the funding from NIH as well as CFAR. And so, yeah, thank you for your attention, and I'd be happy to take any questions.
Well, we're out of time, but if there are any pressing questions, maybe one or two very pressing questions for Sheetal?
Online, so do you want me to ask that for you?
Please.
Did you also quantify the HIV1 GFP positive neurons in the neuronal microglial co-cultures?
So the HIV doesn't infect neurons because, so at least in our model, we were making these co-cultures with infected microglia, because naturally HIV doesn't infect neurons because they don't have this necessary receptors for the infection. So the major cell types that get infected in the brain are microglia and some perivascular or border macrophages. And astrocytes and neurons cannot support HIV infections. So we do not look at that.
Any other questions? Okay, in that case, I'd like to thank you very much for your very interesting presentation, and thank you very much to the speakers this morning.